maybe more. Depends. I cannot give too many because I have a lecture.
Still doesn't work.
Yeah, I just today one day, yeah. Try it. Okay, let's here, the second part is all specific polysaccharide. Does it work now? No. Might? No? Yeah, it looks like there's a signal, but hello? Where's the bottom? I don't think there's a bottom.
Not on that.
Oh. Hello? 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 Is it working? Yeah, it's okay. Okay. Hello? Hello? All right. The all-specific polysaccharide. This is a part of the lipopolysaccharide, and its function is a somatic antigen. Okay. And sometimes we also call all antigen because of the same all antigen. And for identification, and I can show you that. See, this is a the lipopolysaccharide. This is a lipid portion. And the particular lipid A, which contributes to the toxicity of gram-negative bacteria, which can cause fever. All right, and this is a polysaccharide portion. And polysaccharide has two segments here. One is a core polysaccharide. This one is very similar. This portion is very similar among different gram-negative bacteria. But this portion, all specific, as the word indicates, they are very specific to different species of gram-negative. Therefore, it functions as antigen. This portion is antigen, so we call somatic antigen. What does the somatic mean? Soma, body. So the body of the bacteria, okay, the antigen of the body of the bacteria, okay. Or called all antigen sentence, all right? And I also want you to notice that here, do you see that in the core polysaccharide there are lots of phosphate, and also lipid also have lots of phosphate. So this contributes to what charge? Negative, very negative. So all the gram-negative shows a negative charge. Okay, in a gram-positive, it's also negative charge, but the negativity is also provided by the phosphate group of which component? Last time I mentioned it. Anyone still remember that? Teichoic acid. Okay, teichoic acid also has lots of phosphate. Okay, also negative. All right. So both contribute to the negativity.
Say that again.
Both gram-positive, gram-negative contribute to negativity on the surface, cell surface. All right. So here it can stimulate our body, produce antibody, has negative charge, regulate, and this negative charge can regulate ion passage. So it's analogous to teichoic acid in gram-positive. So these two. In gram-positive teichoic acid, in gram-negative is these polysaccharide or lipopolysaccharide. Now, there is one term related to the cell wall called L forms. All right, and this has nothing to do with the chemistry, the L form of chemistry, like the L form of amino acids. Has nothing to do with it. Okay. This actually is named after the Lister Institute, L, because it was discovered in 1935, so-called L Lister. What are they? Simple definition. It is a wall-less without wall or wall-deficient bacterium. So L form is bacterium without cell wall, or with what? Very little cell wall. Retained, okay. And some of L form lose ability to synthesize cell wall. If all peptidoglycan is lost, then they are not able to synthesize new cell wall. But if they still retain residual peptidoglycan, then they are able to synthesize the new peptidoglycan or new cell wall. Okay, this is the side. The peptidoglycan cannot convert it because of the synthesis of cell wall require pre-existing peptidoglycan, so you need some there. Okay, this is some. B, the second, the L form. There are two different types of L form. One is a stable L form, and these are the bacteria completely loses ability to synthesize cell wall. That means that what? They lose completely the peptidoglycan. There's no peptidoglycan here. So they are not able to resynthesize it. Okay. The second type is called revertible L form. They are bacterial. They retain some cell wall component, like the peptidoglycan. So they can reform cell wall. Okay. And for example, stopping antibiotic treatment early may cause revertible L form to resynthesize cell wall and may cause infection again. So what it means is this. Suppose the bacteria cause infection in your body, and if you give the penicillin, okay, and as I mentioned before, penicillin inhibits peptidoglycan synthesis. Remember that? All right. In that case, they will lose the peptidoglycan, and many will die, but some will survive. Okay, even lose the peptidoglycan, they will survive. Okay. Those survival one, okay, now are they sensitive to the penicillin? No, because they don't have a peptidoglycan, a very small amount. But they somehow they survive it. Okay. And in this case, many people thought that the symptom will reduce because many already died, only some survived. So the symptom decrease, and the patient may think that they are cured. So they stop taking penicillin. Once they stop taking penicillin, those survival one, particularly which still have some peptidoglycan, then they are able to resynthesize. You see that? And then multiply very fast. That's the reason. Have you ever experienced that you have infection and after taking a penicillin and improve and you stop and then after a week they come back again? Have you ever heard that kind of thing? It's possible because of this. Okay, they regain the ability to synthesize peptidoglycan. Okay, in the absence of penicillin. All right. The formation of L form bacteria may be due to an overuse of antibiotics, which inhibit the synthesis of peptidoglycan, which is the reason I just mentioned it. And it will remove peptidoglycan layer and it become L form. But some L form can still survive in our isotonic body fluid. And further, same antibiotic treatment will become inefficient, okay, because they don't have peptidoglycan or lose lots of peptidoglycan already. So symptoms decreases, and then after stopping antibiotic treatment, those in L form can revert back to the original form, which I just mentioned, and cause the infection again. All right, another term, protoplast. What is protoplast? They are the bacteria without cell wall. Okay, but this is artificial. It was produced artificial in isotonic solution. If the cell wall of the gram-positive bacteria is completely removed by treatment with a lysozyme, and remember, how does a lysozyme work on peptidoglycan? Anyone still remember? It inhibits beta-1,4 linkage of peptidoglycan, GM. Okay, so if you treat with a lysozyme, then the peptidoglycan will be removed. Okay, or you can treat with penicillin, which is block the peptidoglycan synthesis. Then the resulting remaining wall-less, osmotically sensitive, spherical round cell now is called protoplast. How come they do not burst? Yeah, because in isotonic solution, so they will not burst because outside also high concentration. In this case, that bacteria without cell wall is called protoplast. So I want you to know, when you study, you got to know, ask yourself what it is. So when you see the protoplast, they are what? Bacteria without cell wall. Okay, remove the artificial.
If it has no cell wall, is it not causing symptoms anymore?
No, not necessarily. It depends.
Okay.
It depends, yes. Spheroplast. Okay, spheroplast, very similar to protoplast. The same treatment, okay, in isotonic solution, lysozyme and penicillin on gram-negative. Now this one's gram-negative bacteria will generate wall-deficient spherical cell called spheroplast. Don't forget, they have much less peptidoglycan. However, they have other components of the cell wall. Remember, outer membrane? Okay, and the lysozyme and the penicillin will not destroy that. So they will retain part of the outer membrane, okay, which will retain much of the outer membrane. But if the remaining outer membrane in the spheroplast is removed, you can remove with a special reagent. Then it will become protoplast. So the difference between protoplast and spheroplast, the spheroplast still retains some cell wall component, like the outer membrane, particularly from gram-negative. But you are able to remove specifically with some reagent. And if all the cell wall component, including the outer membrane, is removed, then the resulting cell now is called protoplast. So protoplast simply means that the bacterial cell without cell wall completely. Okay. Spheroplast are bacterial cells with what? Residual cell wall component remaining. So these are two different. So I want you to know the two differences, and they are bacteria. Okay. All right. Now, next one we are talking about archaea cell wall, which is quite different. They don't have a peptidoglycan, but they have something else. We call pseudomurin. Okay, instead of peptidoglycan there, okay, you can see the diagram there, see that very similar to peptidoglycan. Here you can see the N-acetylglucosamine, which you also find in bacteria. But you don't find the N-acetylmuronic acid. Instead, they have an N-acetyltelosamine uronic acid, very long. Okay, so T. So they GT instead of GN. Okay, but if they don't have M, we don't call it peptidoglycan. We call it pseudomurin. And they also have, see that the side chain peptide, and then they also link it. So very similar to peptidoglycan in the structure, but they are not peptidoglycan. All right, and also another thing I want you to notice is here the link between the G and T is beta-1,3, not beta-1,4. So they are lysozyme resistant, insensitive. So lysozyme will not break it up. Okay, so I want you to know this. Okay, memorize this because of beta-1,3, so cannot be destroyed by penicillin or the lysozyme. Okay. Some archaea cells have other type of cell what we call polysaccharide. Okay. and they don't have a pseudomer. Instead, they have a polysaccharide. And here, for example, like a methanol-sarcina. In this case, they require methane. Because this methanol-sarcina exists in some patients with stomach ulcer. It's called ulcer, containing glucose, glucuronic acid, and glucosamine acetate. All this is sugar, so we call polysaccharide. And but this is not the major. This is not the major cause of the stomach ulcer. The major cause of stomach ulcer is Helicobacter pylori. If you have heard it, we will discuss it in the future. Another genus, Halococcus. Okay, whenever you say halo, that means what? Like a sodium, salt. Okay, so they are extremely halophilic, the salt-loving archaea. They have a sulfated polysaccharide cell wall, and sulfate has a negative charge, okay, which can bind to the sodium in the solution. And actually, the sodium sulfate has stabilized the cell wall of these Halococcus, so they can survive like in Dead Sea. Okay, they can survive, and the salt will not kill them because actually it's stabilized. All right, some archaea cell wall are acylate. We discussed this before. Okay, also found in some bacteria, but mostly acylate are found in archaea. Composed of protein, some composed of protein, some composed of glycoprotein. Okay, depends. And they are sufficiently strong by themselves to withstand osmotic binding. So acylate always form the outermost layer. Okay, and you can see the diagram there. See that we saw that before, found in bacteria, some bacteria, but mainly the archaea. The function of acylate, therefore, one is they serve as a structural reinforcement, make it stronger. Second, it serves as interface between cell and environment, so they can communicate. C, it serves as a selective sieve to allow low molecular weight to go in. D, it can retain protein near cell surface, so it can be used for their nutrient. So you can see all these are beneficial for them. All right, and as you can see, the summer cell, the archaea cell has what do you call these in their membrane? Tetraether. Remember, tetra is a monolayer. Okay, we had that before. All right. So, wall-less archaea without cell wall. Some don't have cell wall, like Mycoplasma. Okay, Mycoplasma is bacteria, not archaea. Okay, but they are very similar to Mycoplasma. By the way, I want you to know that Mycoplasma is the major bacterium without cell wall. Naturally, they don't have cell wall. Okay, they don't have cell wall. And also, they are the smallest bacteria. This is a problem bacteria because it causes many human infections, particularly like pneumonia, meningitis, and many others. So this is a problem.
Bacteria.
Mycoplasma. Okay. Now, these wall-less archaea include a thermoplasma. As the word indicates, they are thermophilic. They grow optimally at 55 degrees centigrade, which is pretty hot, 55. Okay. And acidophilic. The optimal pH is a pH 2. It's very acidic. Okay, and they are involved in the sulfur respiration and generate heat, spontaneous combustion. So actually, some of these pile of these archaea, sometimes they can cause fire by themselves. To produce lots of heat, and there is a wood in there besides the cause fire. The second type of wall-less archaea is ferroplasma. And ferro, what does that mean? Iron. Okay. It's strong acidophile. Look at here. It can produce acidic solution at pH 0. If you put your finger in a pH 0, you know what will happen to your finger, right? They produce it and they grow in the pH 0. All right? But it's not as thermophilic because they grow optimally at 35 degrees. And it can oxidize ferrous. This is a reduced form to the oxidized form. So this is the oxidation reduction reaction. And I mentioned before, whenever oxidation reduction reaction takes place, energy is release. So they can generate energy. And this reaction at the same time produce byproduct, acid, pH 0. And they use CO2 as a carbon source. So they are autotrophs. Right, so they can survive very easily based on the CO2. All right, and some archaea, they have a special component in their cell called lipoglycan. Lipo is a lipid, glycan is a sugar or polysaccharide. So these will form tetraether lipid monolayer membrane with mannose and glucose, which is I just mentioned it previously. Okay, and some archaea cells will also contain glycoprotein. So that varies a lot. Okay. All right, so we finish cell wall. Okay. Now, next structure of bacteria or the prokaryote is the capsule. Okay, capsule, I want you to look at what is capsule. See right here. These are bacteria, and they are surrounded by very thick layers of stuff, which actually is very resistant to capsule. Capsule. All right, and see that is a capsule here, and this is big capsule here. All right. And capsule is a problem for human again, because they resist the phagocytosis by our white blood cell. So, if you see any capsulated bacteria, then you know most of them are pathogenic. Okay. If you remove their capsule, then it's okay. All right. What comprises capsules? Could be either polysaccharide or protein or mixture of both. Okay. And the nature of this capsule is that they are very sticky, like a slime. And so they are tightly organized matrix. They exclude particles such as Indian ink or nigrosin. We are going to do in the experiments in the laboratory to stain it, and you cannot stain it. That's why you can see the difference between the cell and background, and in between you have a capsule. And this capsule firmly attached to cell wall and responsible for pathogenic nature. As I mentioned, it causes resistance to the phagocytosis. All right. And similar to capsules are slime layers. Actually, they are very similar. The only difference in material is more easily deformed. So not as solid as this one. This is more easily deformed and does not exclude particles. That's why it's more difficult to see it, because the stain can go through it and you don't know whether it is or not. Okay. It's loosely attached to and can be lost. All right, the function of capsule and the slime wall. And I want you to memorize that. Okay, first one, it protects bacterial cell from phagocytosis by the phagocytes. B, it prevents the viruses from attaching to the bacteria, so it's good for them. Okay. C, it prevents the drying or desiccation by binding water. So it can survive in dry environment. So again, that's beneficial for them. They can survive in the adverse environment. Okay. D, it promotes attachment of bacteria to any surface and cause infection. Yes?
Are these the functions of both the slime layers and the capsules, or just the slime layers?
Both. Both will do the similar function.
Okay, thank you.
Yeah. Okay, and for example, if it's attached to your teeth surface, then it will lead to what? Cavity. That's how cavity is. Cavity is caused by this capsule. Okay, and I'll figure out something. All right, and if it's attached to your lung, then you can cause it pneumonia and then many other things to start with. Okay, cystic fibrosis. Okay. E, another function, it has charge which prevents cells from aggregating together, so they can spread. F, sometimes is responsible for developing the biofilms. We will discuss this biofilm. Biofilm simply is a layer of bacteria on the surface, particularly like in your teeth. Okay, it forms a layer called biofilm. So biofilm has a matrix and which binds to many bacteria and kind of protect them. The capsules cause the aggregation of the biofilm. And a good example is these two bacteria. I want you to memorize Streptococcus sobrinus and the Streptococcus mutans. they form biofilm on the surface of our teeth, which then results in dental cavity or dental decay. The difference is this Streptococcus sobrinus, it forms the biofilm on the smooth surface of our teeth. On the other hand, the mutants, Streptococcus mutants, they form the biofilm in the fissure crack, okay, crevice of your teeth, like for example between the teeth and the gingiva. Okay. So I want you to memorize these two. So next time, if you have a dental cavity, then you know what bacteria you have. You have these two bacteria. Okay. Well, as a matter of fact, probably all of us have these two bacteria. But if you keep on brushing it and keep in good shape, then they will not cause this problem. But you also know that some people they can eat lots of candy without cavity, right? Some don't eat candy at all and have a lot of cavity. So genetic factor play a very important role. So hopefully you have the good genetics, but unfortunately most people not. All right. Okay, the function. Okay, then the flagella. Another is the Cetus flagella. This is a plural, singular flagella. And let you see this. See that this flagella, long one. Okay, this is a long one. Is flagella. Okay, then.
Dr. Lin, the figures and page numbers are now the same on our slides. Does that mean that?
Yeah, this one is corrected here.
Okay.
If it's not corrected, then not corrected. All right, flagella. Okay, the function. Memorize all the functions. Function is very important. Okay. What is it for? For motility. Motility. And many bacilli and spiral bacteria usually have the flagella or flagella, while cocci rarely do. Okay. And they are only visible with staining under light microscope or no stain under electron microscope. Okay. All right. The flagella are arranged in the following patterns. There are four. One is monotrichous. That means it has a single polar on one end. Let me see. I have. I see that here. This one. Okay, monotrichous. Only one on one end. Okay. Then amphitrichous. Amphibian. Amphi means on both ends. Yes. So one at each end. Do I have the picture there? No. But one end and the other at two ends. Okay. And the third one is a lophotrichous. Lopho means a tuft. Okay, a bunch. Okay. Of flagella at one end, and you can see this one. Yeah, this one. See that? This is a lophotrichous. Okay. Trichous. Okay, trichous means hair. Okay, bunch of hair. And then the paratrichous. Para means around. Okay, and we have this here around. So this one, paratrichous. All right. Now, what comprises flagella, the structure? There are three parts. One is the basal body, hook, and the filaments. And you can see the diagram here. Okay, this is the basal body, all here, which is embedded in the cell wall and cell membrane. Okay, this is called basal body. Okay, see that basal body. And then hook, the green one is a hook. And then filaments. See that filament is this long one there. So there are three portions here. So let's go back. Basal body insert through cell wall and cell membrane with four rings and the middle rod in the gram-negative. So you can see it. See there are four rings in gram-negative. Okay, L ring, P ring, MS ring, C ring. But look at it. In the gram-positive, because gram-positive do not have this outer membrane, so they only have a two ring here. So the only difference is gram-positive two ring, two inner ring, and the gram-negative has a four ring there. And you can also see that there are protein here in the cell membrane where you can do two different types of protein. Okay, it's a function as a motor. Okay, it can switch. Okay. And this one is free motor. It's a motor switch. They can reverse and go the other way, so they can swing any direction or backward. It's all this. Okay. So that you can see the more protein everything there. All right. So hook filaments composed of a polymerized flagellum, it's a protein. The mechanics involved by the flagella. Okay. The mock protein is a motor surrounded the inner ring and anchor in the cell membrane, I just show you. And flea protein function as a motor switch in reverse direction of rotation of flagella. So mainly the flagellum is for motility. Okay, and they can swing around. And so it's kind of what? Analogous to our leg and hands for movement. And one phenomenon is very interesting. You can shake the suspension of bacteria, shake it up really hard. Then all the flagellum will be cut off. And then just within a few minutes, they grow back. And then moving again. So hopefully in the future, they can put that kind of gene in humans. We lose our hand, we can grow back. Theoretically, it's possible. The bacteria is amazing. Yes.
So only gram-positive bacteria has the 2 bottom rings.
That's right.
But gram-negative has
4. That's right. All right. The flea protein. Okay. So, okay. And another very unusual phenomenon of flagellum. They're very powerful. Okay. They can move and they're very powerful. How fast? Look at it. They can rotate it up to 300 revolutions per second. What does that mean? That means it can move through the liquid at 60 cells length per cell. Wow. By contrast, the cheetah move only at maximum rate of 25 body length per second. So you can see actually what inch 4 inch what actually these guys the flagella is much more powerful, very very fast. You can see it. All right. Now there's one type of motility is not caused by flagellum. Okay, we call gliding motility. It occurs in some prokaryotes without, so look at the notice, without flagellum. It is a type of motility. It's a slower, smoother form of movement, typically along the long axis of the cell. Sometimes cells glide by secreting a polysaccharide slime, which contact the cell surface and the solid surface. So what is the difference between the gliding motility and the motility caused by flagellum? The motility caused by flagellum, they can change direction and stop and then go and stop and then change direction. But in the gliding motility, usually what? Go one direction, that's it. Okay? All right, next one. Next structure, fimbriae, very similar to flagellum. Okay, and let's go back to. Okay. Where is the fimbriae? Oh, one more. Yeah. See that here? The long one is flagella, but those are very short and straight here. Those are called fimbriae. Fimbriae, it's a Latin, means finger. Okay, so fimbriae, very short here, all around body, more numerous, not more than the flagella. It's a filamentous structure. Okay, what is it for? All right. Number one, it enables organisms to stick to the surface, including animal, for colonization. So it's very similar to all the one function of what? Which also attached to the surface of your teeth. What is that? What do you call it? The capsule, right? Okay, so it also has a similar function as a capsule. Okay, so this also contributes to the biofilm. So biofilm not only caused by capsule, but also can be caused by fimbriae. Okay, and some flagella actually also can contribute to the formation of the biofilm. And it forms thin layer of cell on liquid surface, and this is called pellicle. In your laboratory, you'll find it out in bacterial growth and broth. Actually, they on the top. Okay, when you stay for a while, you can see a layer of cell we call pellicle. Okay, which is a thin layer and this is caused by fimbriae. Number 3, function. It forms thick layers of cell called biofilm. So I mentioned it just like capsule, flagella. Okay, so these 3 can form it, the biofilm. So I want you to know capsule, flagella, fimbriae all can contribute to the formation of biofilm. Okay, and make sure you read this section. It's very important because this biofilm is now $1 billion business in the whole world. It causes a lot of problems. Okay, we'll talk about that now. It attaches to the habitat and do not wash away from favorable environment, so that they can survive easily, and the water cannot not flush it at all. And it becomes 1,000 times more resistant to antibiotics because what? It's a layer of the matrix to protect the bacteria inside, so the antibiotics cannot easily penetrate, or chemical cannot easily penetrate. There's a problem. Antimicrobial drop because of a slower growth rate in biofilm and a reduced penetration of antimicrobial substance through the biofilm. So it's very hard to kill them. Okay, it's very hard to kill them. All three contribute, okay, to the pathogenicity of bacteria, including Salmonella, which causes salmonellosis, some food poisoning things, and gonorrhea, Bordetella pertussis, whooping cough. Now, biofilm. It's assemblage of bacterial cell attached to a surface. Could be the human, could be animal, could be any surface, like a pipeline. Okay. And enclosed in an adhesive matrix because they secrete substance, very adhesive. Okay, and they can trap nutrient, so biofilm trap the nutrient for their growth, help prevent the detachment of cell on the surface. Okay, and what caused the formation of biofilm? So far, we find it out there are several molecules can give the signal for the biofilm formation. And particularly one is called cyclic dimeric guanosine monophosphate, CDI-GMP, and acylated homoserine lactone, and there are many other things. They are still doing the research. Will signal biofilm to form and decrease the flagella. Flagellar motility to become stable, so they will stay in one place. All right, and among all the bacteria, the most notorious biofilm former is Pseudomonas aeruginosa. Memorize, because this causes human disease and lots of problems. They are the most notorious biofilm former. Okay. By the way, the one you are going to do in the laboratory is not the pathogenic one, it's non-pathogenic, so don't worry about it. It forms biofilm in human lungs, causing cystic fibrosis. Okay, the pathogenic one. And the signaling molecule is acylated homoserine lactone. They can produce it and stimulate for it. Okay. All right, now, why do biofilm form in a bacteria? There are several reasons. One is a microbial self-defense, so they are protected. Okay. Number two, it allows cells to remain in favorable niche. Okay, they can find the best place for survival and then stay there and form the biofilm. Number three, allow bacterial cells to live in close association with each other. So it contributes to the bacterial community. So not only human form the community, bacteria forms community because they help each other to increase the chances for survival. And this is a typical way of bacterial cell growing in nature. All right, so we have to control the biofilm formation. That's why I say it's $1 billion business every year. Because this biofilm has a significant implication in human medicine and commerce and also in industry. For example, in human, they are responsible for causing the periodontal disease, for example, like a cavity, I just mentioned it, okay? And if it occurs, forms in the kidney, then it can form the kidney stones. Okay, and also responsible for the tuberculosis, okay, Legionnaire disease, and the Staphylococcus infection, and all you can see that this is a serious problem. So medical implants are also an ideal places for biofilm formation, which afflict about 10 million people per year in USA. So any implant is a good niche for the formation of biofilm. So that's why all these implants has a lifespan. Okay, like the artificial joint, you can only use about 10 years. That's it, and then you have to replace it. Nothing we can do about it. Okay, so it's a problem. And not only that, in the industry, offshore oil rig underneath all form the biofilm and then cause corrosion. So after a while, you have to rebuild it, and it's very expensive. Okay. Another thing is, particularly it's significant to us, is water pipeline. The water we drink, okay? You know most of the water pipeline in the United States is more than 100 years. If you open it up, you will see there was biofilm, larger biofilm inside. So they slow the flow of the water. Fortunately, most of bacteria in that water pipeline are non-pathogenic. But who knows? If some terrorists try to induce some pathogenic there and form their own biofilm, then we are doomed. Then we cannot drink. Does it make sense to you? Now, the terror group actually are working on this biofilm. Okay, they try to see how they can introduce it through all the water pipeline. Okay. All right, and all the boats underneath. Have you seen the boats underneath when they lift up? All the stuff was a biofilm. So they will cause corrosion. So it really caused a big damage. And now the industry try to figure out, see whether they can get rid of it. So far, no success. Okay? I don't know whether we'll be successful or not. Probably until the Lord come back. Because that's too large problem for us to solve. We only know that one of the chemical called furanols. I want to memorize furanol. It is a chemical can prevent biofilm formation. Not absolute, but it to some extent. And fortunately, these furanols are stable and relatively non-toxic. Okay. But now we also find it out in the experiments, the bacteria develop resistance. So we have to look for other substances. Okay. You know, sometimes we've thought that humans are the smartest creature in the world. I don't think so. The bacteria, they know how to develop the resistant things. I don't know how they figure it out. They don't take the physiology, they don't take chemistry at all. They don't know how to do it. All right. So again, what are the two bacteria cause your dental decay or cavity? Streptococcus, Sobrinus, and mutant. All right, memorize it. Okay. What structure contribute to the biofilm? Capsule, fimbriae, and flagella. Okay. All right. So don't forget it. What substance is known to prevent the formation of biofilm? Okay. Things like this. Okay. As you study through it, make sure you memorize it and memorize all those functions. Okay. All right. So next time, you know, when people have those implants, and there's a problem, biofilm, or even the one simple one like a urinary catheter, they can also form biofilm and cause infection. It will be there for a while. All right. Next one. Fimbriae. Oh no, we are staying fimbriae. Fimbriae are different from flagella because from the picture I show you, and I want to know the difference. Number one, fimbriae are shorter, as I show you that in the picture, finer, and more numerous. Okay. Second difference is, fimbriae do not have regular wave appearance, so straight. But flagella is like a wavy form. Number three difference. Fimbriae are not usually associated with motility, but that is not absolute. But usually it's not. It's for other purposes, like biofilm. Exactly. Okay. For the motility, which one contributes to it mainly? Flagella. Okay. Number four. Fimbriae can only be seen by electron microscope because they are so tiny and you cannot see it with the light microscope. But flagella can be seen with light microscope. That's the main difference with a spatial stain. All right. Next structure of bacteria called pili. There's a plural. Singular is pilus. and let's just see the picture. Okay, see that? Okay, first, what are these? The very short one, what do you call this? Fimbri, that's right. Now, this one is very long, okay, very long one. And actually, one of the functions is to attach to another bacterial cell, okay, and then bring it together into direct contact so that they can transfer DNA between the two cells for the genetic purpose. Okay. In other words, that we call pilus. And pilus actually have a receptor for many viruses. So in order to see it under microscope, okay, we put the virus there and then stain with it, and you can see it. If you see the virus there, with the staining, then you know there's a there. So it's longer, usually gram-negative bacterial have pilus. Okay, most gram-positive. It has receptor for binding viruses, very long, and usually contain 1 or 2 pili per bacterial cell. Only 1 or 2. But how many fimbri per cell? Oh, a lot. Nobody knows exactly how many. Function of pili. First one is for colonization. Same thing, and stick to the surface. So same function as capsule, fimbri, flagella. Okay, so they kind of have many same function. Okay, number 2. Some can perform twitching motility, another type of motility, but not the flagella cause motility, twitching, which is a type of gliding motility across whole system, like the one we just showed you last time. Okay. Number 3. Immediate, number 3 is the most important function. Okay. Immediate genetic transfer by conjugation. Conjugation means what? The direct physical contact between two cells. So, pili, it will stretch it out and then attach to the gram-positive, gram-negative. So in other words, they know which one. So in other words, what? One is male, one is female. They know which one. Even they don't have eyes, they know where to touch it. And then put the other one into the direct contact, and then they can transfer DNA. We will discuss these in the genetic portion because that lead to the formation of genetic engineering. Okay. The development of it. All right. Next one is endospores. Okay, and you can see the endospore, see that here, and you are going to do that, observe it in the laboratory. So this bacterial cell, and after spatial staining, you can see that there is unstained area or some stain, it depends on different color, okay, that is like green. Okay. These are the spore, okay, and these are spores. Some spore are at the end, we call terminal. Some at the subterminal, somewhere around there, and some outside. See that is outside. Okay, so central spore, okay, and then the subterminal, okay, terminal. So endospore can be at the end of the cell or the center. And usually 1 spore per bacterial cell, usually. Sometimes you can see 2, but the one you are going to do the experiment is only 1. The dormant of or resting phase of bacterial cell. So, what is endospore? Endospore represents the dormant or resting phase of bacteria, kind of similar to seeds of plants. Okay, and about 20 different genera of gram-positive bacteria are capable of forming endospores. However, no archaea form endospores so far. Okay. And these endospores actually are very resistant to adverse environmental situations like heat, chemical, UV light. So they can survive. as a matter of fact, recently they have discovered a spore in Egypt, dated about 3,500 years back, so about the time of Moses. And they induced that spore to grow back into bacteria. So this is one way they can survive for a very, very long time. Okay? And this also causes a problem in humans. And you can see that this is a vegetative. Whenever you see the word vegetative cell, that means an active growing bacterial cell. Active growing bacterial cell. Okay, so normal bacterial cell. And when the environmental nutrients become deficient, okay, then they got the signal, okay, to form the spore so that they can survive in the absence of nutrients. All right, so they form the spore, and then spore then can release from the bacteria, and this one can survive many, many years. Okay. All right, so you can see the spore here. As I mentioned, there are about 20 different genera of bacteria are capable of forming endospore, but there are three are the most important. Because with respect to the clinical situation, okay, because they cause a problem. Okay, so 3 examples of gram-positive format. The endospore structure. Okay, from the innermost to outermost layer. We'll discuss it in a minute. Okay. 3 examples. Let me show you the first 3 examples. Do I talk about that 3 examples? Where is it?
You have it in our slides.
Oh, yeah. Oh, yeah. All right, bacillus. Okay, and the bacillus is aerobic and usually pathogenic. For example, like Bacillus anthracis, which causes anthrax. I think I put it there. How come it's not here? Do you have it? How come I don't have it here? Okay. All right. You have a newer one than I have. Okay. So like anthraxes, they cause a problem, and many others. Okay. And another one is a Clostridium. Okay. These probably the one of the worst bacteria. So many diseases are caused by it. Okay. And whenever you see the Clostridium, you better stay away from it if possible. Okay. They are what? Gram-positive. Okay. And they are anaerobic. You have that anaerobic in your slide. Okay. They are anaerobic. And they are responsible for many diseases. For example, like one type of Clostridium causes tetanus. Another type causes what? Botulinum. Clostridium botulinum. Okay. Another one causes the Clostridium perfringens, which cause food poisoning. In your the food poisoning. There are many others. Okay. And as you can see, tetanus is serious. Okay. Think about those. Okay. Another one is Sporosacina, the carcass shape found in soil and decomposed of high level urea. So raise pH of soil and act as a fertilizer. So this is important for the agriculture. Let's see.
When it said photogenic, it said on the slide you were just talking about, it said photogenic. What is that right?
No. That's a misspelling. Pathogenic. I think I corrected, didn't I?
When you said it, you did.
Yeah, the revised one. Did you download revised one?
The advanced one.
Yeah, pathogenic. I already corrected. Okay. So make sure that each time before the lecture, I'll upload into that. Okay, probably the night before. Okay, make sure to check it and just re-download. Okay. Okay. So you don't have to rewrite it. Write it for you. Okay. Any question up to this point? Any questions up to this point? All right, and now let's get this solved because it's already, my throat is already dry. Okay, the endospore structure from innermost to outermost layer. Okay, there is a correction from your textbook. Your textbook actually is not correct. Okay. So I corrected that. Okay, the first. Okay, this is the core, and core is the future cell. Okay, this core just like an ordinary cell. It contains DNA, ribosome, those organelles. Okay, they're all contained here. Okay, so we call core. Okay, it's a major portion. Okay, and then outside the core is a core wall or spore wall. So in other words, let me explain this a little bit. Right, this is cell membrane. And when the situation is optimal, that means not enough nutrient, then they'll give a signal. So then they will invaginate it. All right, and they keep invaginating. Okay. And then it will fold it. And so look at here, you have a double membrane. It's a one membrane. You see it? And then they will fold it together and it forms something like this. The two membrane. So now they have a two membrane here. And then they will synthesize. Okay, let me exaggerate. Okay, this is the inner membrane, outer membrane, the cell membrane. Then they will start synthesize the new layer. We call it core wall or spore wall. So it's outside the inner membrane there. Okay, and it contains normal peptidoglycan and will becomes the cell wall of the future bacterial or the germinating cell. Okay. Now the second. Then it will form cortex. Okay, this is a cortex. This is a core wall, a spore wall. And this is the thickest one, the cortex. The cortex is the thickest layer, contains loosely linked peptidoglycan, and also contains a very unusual chemical called calcium dipicolinic acid, or DPA, which is in the diagram. Let me show you here. Huh? Where is that diagram? Where? I know that I put the diagram there. Do you see the diagram of that picolinic acid? Yeah, this is the old one. Okay. Strange. I didn't put it in one. Okay. All right. If you have a new one, the revised one, you should have that together. I believe it's together with one of these pictures. Is it one with this picture?
It's on the bottom.
It's there, right? Yeah. Like I put it underneath here. Yeah. Okay. And you can see it has a negative charge which binds to calcium. And this picolinic acid with calcium actually can reduce the water, so make it dry, so they can survive in the dry situation. Okay. All right, so this is a cortex. And And this cortex is extremely sensitive to autolysine. Extremely sensitive to autolysine. Remember, what is autolysine for? I had that last time. Anyone remember what is autolysine for?
Forming the breaking or the transpeptidation.
That's right. In other words, what? The bacteria will grow, right? And if they grow, they have to increase the size, correct? So that means what? They have to break up their existing peptidoglycan first. Remember, it is the function of autolysine. Then the new peptidoglycan component can add to it and then linked again. Okay. All right. So this one is extremely sensitive to autolysine, and will be broken up first in germination. Okay. So actually, when the time comes and they will transforming back into the bacteria, then this the form become active first. Okay. Cortex. Okay, and then next one. That next one, it will forms the spore coat. Okay, this is spore coat. And at the same time, this outer cell membrane then disappears. Okay. And spore coat is one of the strongest layer. Okay, and I'll see why is it here. This as outer cell membrane disappears, and other layer form called spore coat. It composed of a spore-specific protein similar to keratin. Where do you find keratin? Your hair, your nail. That's why it's tough. Okay, so this is why it's so tough. It resists to heat. Chemicals contain high concentration of SASP, small acid soluble protein. Okay, we'll discuss this in okay. And then finally, they form the last layer. It's called exospolium. So this is exospolium. Exospolium. What is it? Okay, exospolium is outermost layer, and they are thin protein cover. Simply, it's a thin protein covering. Okay. All right. Next one, we are going to discuss the function. Okay. We'll discuss it. So that's okay. Today, let's stop it here. Yeah.